Hi there and welcome to this virtual revision guide for Unit 2 GCSE Biology. And today we're going to quickly go through the main aspects of this module. And just to remind you that this is not enough to get you the strongest marks, it's just to make sure you have the basic understanding that you can build on yourself in your own revision and independent work. So we start this module with the area of organisation, and organisation shows us how a living thing moves from being the smallest part of us, which is a cell, up to the whole organism level. And we basically go across several different levels from A, the cellular level, which in this example is a heart cell, or a muscle cell of a heart, to a tissue level, which in this case is a muscle, to the organ level, which in this case is the heart. Then the heart is part of the organ system, the circulatory system, and that's part of the whole organism. Now, in terms of learning the definitions, you do need to know what a cell is. It is the basic functional unit of a living organism. So basic functional unit is fine, the basic living unit is fine, but it's the smallest living thing there is. Then a tissue is a group of cells working together to perform a particular function. So for example, a muscle tissue is a load of muscle cells working together for a particular function. And then that sort of definition continues then. An organ is made up of a group of tissues working together for a particular function, and an organ system is made up of a group of organs working together for a particular function in the organism. Now, there are three types of tissues that you need to be most aware of. The three types of tissues are muscular tissues, glandular tissues, and epithelial tissues. And I'll often ask you to give an example of a tissue that is in the stomach, and all three of these are found in the stomach. You might be asked what they do particularly. So a muscle tissue is able to contract, and that brings around movement, so the stomach moves to churn the food. Glandular tissue is used to release hormones or enzymes. So in the stomach, enzymes are released to help digest the food. And epithelial tissues line parts of the body or cover parts of the body. So in the stomach, that's important because it stops the acid digesting the stomach. But also you'll know about ciliated epithelium in the breathing system. And the alveoli are also covered in epithelial cells. So one part of living is important is diffusion, and you should really know from earlier on in school that diffusion, which you can see in this diagram, is the movement of fluids. So it's either a liquid or a gas, not a solid. So liquid or gas is a fluid. And it is the movement of particles from an area of higher concentration to an area of lower concentration. And this happens due to the random movement of particles. So they collide with each other, and that causes them to spread out and from a higher concentration to a lower concentration until they are completely spread out. And it is a passive process, therefore it doesn't require energy. Now, whilst we say it is passive, energy will obviously help, and therefore the things that can affect the rate of diffusion include temperature. So the higher the temperature, the more energy the particles will have, and therefore the quicker they'll spread out. The size of the molecule, so the smaller or lighter the molecule, the quicker it will spread out. The distance it's got to travel, obviously the shorter the diffusion distance, the quicker it will move there. The concentration gradient, so if it goes from a really high level to a lower concentration, it will do it quicker than if it goes from a relatively low to a bit more lower concentration. And the surface area, which is why things like alveoli have a large surface area. And you can see here what happens is that, let's say oxygen, for example, high concentrations outside the cell diffuse into the cell. And because it's diffusing into the cell, then it will keep diffusing until there's an even spread inside and outside. But obviously in real life, the cells are using up the molecules, so that is less likely to happen. Now, we mentioned oxygen in the last slide, and we said about our gas exchange in the alveoli. We need to know how we actually get that gas and that oxygen into our blood cells. So we do this obviously through breathing, and that gets the oxygen in for us and gets the carbon dioxide out. But obviously it's air that's coming in and air that's coming out. So when we breathe in, and you need to know the basic structure of the breathing system, the respiratory system, and obviously the air comes through the mouth, down through the protected trachea, into the bronchus, into the bronchioles, and into the alveoli. Now, when we breathe in, there's a pressure change that happens, and you need to know the stages, and often an area that people don't learn very well. So first thing happens is when we breathe in, the muscles called the intercostal muscles contract between your ribs. This causes the ribs to move up and out. The diaphragm then, which is this dome-shaped muscle underneath your lungs, the bottom of your thoracic cavity, the diaphragm contracts and this flattens it. This causes the volume in your thoracic cavity or your chest to increase, and therefore the pressure decreases and air is pushed into the mouth because the pressure is lower than that of the atmosphere. When you breathe out, your muscles relax, so they do the exact opposite from what we've just said for most of these parts now. So if you learn one, you've automatically got the other one. This means the ribs move back inwards and back down. The diaphragm relaxes and it causes this dome shape to appear, so it pushes upwards on the lungs. The pressure in the chest decreases, so the volume on the chest decreases, and that causes the pressure to increase, which forces the air out because this pushes the air out because it's got a higher pressure than the atmosphere. Now when it gets to the alveoli, there are 4 adaptations, which are key 4 adaptations that come up a lot in biology that you need to know about exchange surfaces. So whilst this is about the alveoli, which is part of the human lung system, I've seen questions that link this to the digestive system and the villi in the digestive system. I've seen questions that link this to gills in fish and to external gills in axolotls. So they are adapted to allow the diffusion of oxygen into the blood and carbon dioxide out of the blood by having the following 4 adaptations. They have moist walls, which helps diffusion happen. They give the lungs a really large surface area, so therefore lots of molecules can diffuse out in one go. They have lots of tiny blood vessels, or good blood supply as we call it, that maintains the diffusion gradient of high to low, and they are only one cell thick, so there's a short diffusion distance for gases to travel. So another type of movement or process of movement in the body that we use is something called active transport. And this is where, opposite from diffusion really, molecules move from concentration of a lower concentration to an area of a higher concentration. And this uses energy, or sometimes known as ATP. So if you were asked to define it, you'd say the movement of molecules from an area of lower concentration to an area of higher concentration using energy to do so. And what the energy does is it changes the shape of something called transport proteins or carrier proteins on cell membranes. And basically what they do is they change shapes. You can see here molecules go in and they're in a region of lower concentration. The shape changes and that releases the molecules into the area of higher concentration. And this happens in a couple of key areas. One area you need to know about is it happens in the digestion of glucose in the human body, in the intestines, and it also happens in the root hair cells. So if you are asked for adaptations of the root hair cells, the adaptations of the root hair cells are pretty much what we looked at in the opposite slide. So they've got a large surface area to allow rapid movement in of many molecules. They've got thin, so they only allow one cell thick, so it allows a short diffusion distance, but also they have high levels of mitochondria in the root hair cell. Why would you have mitochondria? Because the mitochondria allow high rates of respiration, which releases energy to allow active transport to move molecules from a lower concentration to a higher concentration. Then we move on to the digestive system. So digestion is the breakdown of large insoluble molecules into small soluble molecules. So what that means is we're taking big molecules, breaking them into small molecules, and we're turning insoluble, things that can't dissolve, into soluble, things that can dissolve. And the reason we do that is because we want those things to be able to move around our bloodstream. And that's the whole point of digestion, is so they can enter the bloodstream. So this second sentence here is vital not to overlook, because that's the whole purpose of digestion. And we need to know what different bits of the digestive system do. So the digestive system, the mouth starts the digestion with an enzyme called amylase, which digests starch into glucose, and we also chew the food and manipulate it with our tongues, and that breaks the food into smaller bits. The stomach is where main protein digestion happens, and there's acid in the stomach, hydrochloric acid, which kills bacteria and other pathogens that could potentially make us ill. There is the small intestine where fats or lipids are digested and we absorb that digested food. there is the large intestine where water is absorbed, and then storage happens in the rectum. Now, in order to digest food, we need to use something called enzymes, which are biological catalysts that speed up the rate of reaction. So enzymes are not just for digestion, they're used for lots of reactions, and digestion is one of those types of reactions. They are always made of proteins, and they have this basic structure here, which is a typical drawing you might see. The key part of the enzyme is the active site, which is this bit here, and it has a specific shape that is complementary to the thing it's trying to react with. So we call the thing that is reacting in the enzyme the substrate. So for example, if this was digestion, you might break down a substrate of protein, and that would be broken down into amino acids here. And the only enzyme that can break down protein is the enzyme protease, because it has a specifically shaped active site for the protein to fit in. So the active site is complementary in shape to the substrate, and that makes them specific. They'll only break down, in this case with digestive enzymes, one thing. This is called the lock and key model, because the active site is specifically shaped, it's complementary to the substrate. Now, in enzymes that digest, we have three main groups of enzymes that you need to know for GCSE. So the first group we're going to talk about is in the mouth, and that's the first enzyme that happens in your saliva that comes out, and that is carbohydrase. And carbohydrases are also, you might be told, amylase. You could use any of the names below, but this is the one that will be in the GCSE books. So you'll either call it carbohydrase or amylase, and this breaks down carbohydrates such as starch into glucose. So starch is a long chain of glucose molecules, and then it's been broken down into individual molecules of glucose, so it's smaller, and glucose is soluble, so now it can flow into the bloodstream. In the next part, we have the stomach. The stomach is where proteins are digested. So an example of a protease enzyme is pepsin. And again, it breaks down large insoluble proteins into small soluble amino acids. So that's the digestion of proteins. We then have in the small intestines the lipids or fats, and they get broken down by an enzyme called lipase. And lipase breaks down the large insoluble fats into smaller molecules called fatty acids and glycerol. Now, the pH of these enzymes is very important. They have to have a specific pH called the optimum pH. So you also need to know that in the mouth the pH is pH 7, which is perfect for the enzyme amylase. In the stomach, because of the hydrochloric acid in the stomach, the pH is 2, but that is the optimum pH for protease or pepsin. And in the small intestines, the pH is about 8, and that is optimum for the enzyme lipase. Now, to get that pH in the small intestines, we have to use a chemical called bile. And this is because the food is coming out of the stomach, which has got pH 2, and if it got into the small intestines at pH 2, it would denature or stop working the lipase enzymes. So the pH has to be neutralized and taken up to pH 8 from the highly acidic pH 2. And the enzyme that does this is bile. So non-enzyme, the chemical that does this is bile. So often you might get asked about the digestion of fats in a bigger question. And what you would say is the digestion of fats occurs due to the enzyme lipase, which breaks down the fats into fatty acids and glycerol. But to get the pH correct, pH 8 for this lipase to work, we also need the non-enzyme bile. And bile does two things. It neutralizes the stomach acid, which is already said, and it also emulsifies the fats. And what this means is it basically takes a big lump of fat and breaks it into small droplets. So then these small droplets have a large surface area and can be readily digested via the lipase enzyme. But it's important to say that bile doesn't digest, it doesn't do any chemical changes, it just takes big lumps of fat into small lumps of fat. And I think it's also worth saying that all of these enzymes, so amylase, protease and lipase, are all produced here also in the pancreas. So enzymes have a specific set of conditions they like, and one of those is having a pH that is very specific. So enzymes work at optimum pH. If the pH is wrong, what happens is the shape of the enzyme and the shape of the enzyme's active site changes shape, and we call this denatures the enzyme. So the enzyme doesn't work at the wrong pH. So you can't say kills the enzyme because the enzyme is not alive, but it denatures it. And if the active site changes shape, it is no longer complementary to the substrate will no longer fit in, and the enzyme substrate complex can't form, so the reaction can't happen. Now, if you were to look at the digestive system based on what I've just gone through with you, you might get a graph that looks something like this and be asked to identify the enzymes in an exam. So enzyme A here has an optimum, if you look at the graph, of about pH 2. And therefore enzyme A would have to be the one in the stomach, which is the pepsin or the protease which digests in the stomach because of the stomach acid. Enzyme B here has an optimum of just around about pH 7ish, just a bit below. So that would be the carbohydrates or the amylase, which would be in the mouth, and therefore it's got a pH above that. And then enzyme C has just above pH 8 to pH 9, and therefore enzyme C would have to be the lipase, which works in the small intestines, which has a pH of about 8 following the action of the bile. Temperature is one that is very important. You're often asked about much more. So this graph shows you the effect of temperature on the rate of enzyme activity, and this is for any enzyme, not just digestive enzymes. So here you got given this graph and you were asked, how does temperature affect the rate of reaction in enzymes? You must start at 0 here, because what often people do is they just talk about the above 40 degrees, and that's a mistake. So what you would say is the rate of reaction increases as the temperature increases. So all this bit here, as the temperature goes up, so does the rate of reaction. Now, why does that happen is absolutely critical. So the reason that happens is that the enzymes gain more, and the substrate also gains more kinetic energy. So they're moving around quicker, and as a result, they collide more often, which links to something you'll do in chemistry as well. And because they collide more often, you get more enzyme substrate complexes. At about, in this temperature, about 40, and always read it off the graph, don't assume. So I'm going to say at about 42 degrees, we hit the optimum temperature. And always state in the exam what that is based off the graph. Don't just assume it's 40. So at 42 degrees in this example, we reach the optimum temperature, which is where the most collisions are happening and the most enzyme substrate complexes happen. Above 40 degrees though, there's a massive drop, so always talk about the fact the rate of reaction drops above 40 degrees rapidly, and the reason is because the enzyme does something called denatures, which is on the previous slide. And the enzyme denatures because the active site loses its shape and therefore is no longer complementary to the substrate, so it can no longer catalyze the reaction, so the enzyme is denatured. Now, you also need to be aware of the required practical for this module, which involves food tests. And food tests are hard to remember, but need to be remembered. A very popular exam question. So very, very quickly, here are the following the food tests you need to know. First food test is called the Benedict's test, or the Benedict's reagents test, and you add a couple of drops of Benedict's solution to your food. That is a blue sort of colour, and if you then put that test tube in a water bath for 5 minutes, about 80 degrees C, it will go green, yellowy orange, brick red, or if glucose is present, and it forms a precipitate, so a solid will form in there. Next example is starch. Most people remember well. So you add a couple of drops of iodine to the food. If starch is present, it will go from a yellow-brown colour to a blue-black colour. Protein test is adding something called biuret test. So this is often the one that people confuse with the Benedict's glucose test. So you add a couple of drops of the biuret solution, and then you will watch. So you don't heat it up like in previous examples. This one, it will go purple or lilac if there is protein present. And the last one is the fats or lipids test. And there are actually two tests for this. The main one you're expected to know is the emulsion test though, which is where you add ethanol to your food, shake it, and then add water to your food. And if fat is present, if lipids are present, a white precipitate will appear. There is also another test called the Sudan-free test, which is a red liquid which you add and shake, and if there is a fat or a lipid present, it will appear in a solid sort of precipitate at the top in a red level where the red attaches to the fat. That's been useful, guys. Make sure you learn that effectively, and particularly remember those food tests are part of the required practical, which are important. It's important just to state that all food tests should occur in solution, so they don't really work very well with solids.